Welcome everybody to another episode of the Beyond Terrain podcast. I'm your host, Leah Dalton. Today we're gonna have an amazing episode. This is probably some of the most important work that's going on in real time right now. So I'm really happy to be able to bring this forth to you. We're gonna have a little presentation today. Be best if you could hop over on the YouTube to watch it. I'm sure if you're just on the podcast, you can probably get the gist of it, especially if you know what we're talking about here. The topic is the end of virology, and this is sort of the times that we're in, and I'm sure many of us would agree that virology's already been disproven, but the work's not necessarily done. I think that there's always strength to be added to the argument to make it sort of undeniable and to allow it to gain traction. So this man today, Jamie Andrews, is doing some phenomenal work. So I'm really grateful to have him on today, and I think we're gonna have a really cool, interesting presentation. So I'm really looking forward to it. Jamie, thank you so much for coming on today.
Hi, Lev. How are you doing? Thank you for inviting me on, reaching out to me, and it's great to be able to present the project to you and to all of your viewers. Um, yeah, I've seen you with great interest and watched a lot of your other videos in terms of the terrain and getting yourself out there. So props to you for doing it, and again, thank you for giving me the chance to explain what's going on.
Certainly, yeah, and I'm sure a lot of viewers are already going to be a little familiar with what you're doing, but to get the whole presentation is going to be amazing. So I'm so looking forward to this. This is as much of an episode for me as it is for the viewers. So I asked all my guests at the start, and just before we hop in, I'd love to hear your definition of health. What health means to you, how it manifests, what it looks like.
Health to me is just clean living and no allopathic medicine. Me nor my family, my children, my wife take allopathic medicine at all. We try to heal and detox ourselves, through the symptoms of disease, and we do that, in conjunction with eating healthily, trying to, I have a homestead here, so I grow my vegetables, I grow my fruits myself, and so we look out over the whole easy kind of production chain, in terms of the fact that we know that there are no pesticides on it, we know that there are no agro fertilizers on it, and so on and so forth. So it becomes a whole lot easier to watch your health um, when you take things into your own control. We were talking just before this on just off screen that I've moved into a maison de maître here in the southwest of France, and it's off grid here. So I'm trying to do everything to be self sufficient, and so take on permaculture. Principles of growing food, and that makes it easier to stay healthy, right? Rather than relying on going to the supermarket and checking all of the bad labels on the back. So that's how I kind of manage my health, trying to do it from taking control from the very first bite of food to not restricting your detoxing by taking allopathic medicine that will cover up your symptoms.
Yeah. And it's a lifestyle thing, right? It's a
That's
whole
right.
lifestyle shift. Often that's some of the advice that I try to impart if someone's looking to heal. I think, there's no magic bullets. We know that, whether it be supplements or allopathic medicines,
Correct.
you do have to have a shift in lifestyle. This modern lifestyle is conducive to disease, which is why we see the high rates that we have now, especially living in cities. It's very difficult to be healthy. You have to sort of shift back and reintegrate, relearn what we've known for forever. You know what I mean?
That's it.
It's
That's it.
we've never been this diseased in history, so there's a lot that we can learn from our ancestors. So yeah, no, that's great, and I appreciate that you're you're living the lifestyle. That's great to hear. That's something I aspire to do
Well,
eventually
I'm
too.
trying to
Have
do
my
it
own
in
terrain
glamour.
center.
I'm trying to dissolve the illusion of off grid living, of having a big beard, and you speak to the birds in the trees and they're your only friends, and you're in a log cabin, and I'm trying to do it in style. So I bought a maison de maître, which is like a small chateau, and have built it myself, refurbished it myself, and everything here is built towards being completely independent from the state. So all of my electricity is run off of solar panels. All of my heating in the house is made from sawdust pellets. So I have a wood pellet press off the back of the tractor, and I get the sawdust from a local menuisier wood.
No.
The French is coming in. The like a joiner, a joiner.
Joiner,
I get
yeah,
all my
okay, yeah.
sawdust from a local joiner to make wood pellets to heat the house, collect the rainwater off the roof, and recycle that into using as irrigation on the homestead on the plot. So, trying to pave a way for glamorous off grid living, as it were.
Amazing. I love it. All right,
Cool.
I'm
So
so
do you
eager
want
to
to
hear
do you want to
this.
jump in? I
Yeah,
will.
let's jump in. Pull it up. Yeah,
There you go. Is that coming through on your screen?
coming through all right. Yeah,
Great.
is that perfect? All right, good sir, take it away. It's sort of straight up and down there.
Yeah, that's
Is
right.
that what
I've
it's meant
I've
to?
it's all in portrait. So
Oh, cool. All
I
right, right
just
on then.
because of the way that the slides work, it seemed the best way to do it. I have got slight PTSD from doing doing PowerPoint presentations. I haven't done them since university, and obviously running this project, I needed to put something together in PowerPoint presentations. So it's kind of like
Yeah,
getting the sweats. Like, is somebody going to be marking me down on this or whatever? So
we'll
maybe
go
the
easy on you.
presentation isn't up to kind of professional standards, but
Well, it's nice
we'll
actually the
make do.
portrait mode because we're
It's
all
it's
glued
the
to our
results
phones
that count,
nowadays.
right?
Yeah, that's it. It's the results that count. Perfect. Well, I'll let you take it away here and you just
Cool.
let it flow. And maybe if I
Um,
have a few questions, I can interrupt you if you don't mind.
yeah, I'll just give you a brief, kind of introduction into how I came about running this project. The
Please.
project is a large project. There's almost 100 of us behind the scenes working with countless CROs, contract research organizations, essentially mercenary laboratories that are able to be hired by anyone, by people like me, a complete layman, and a complete I knew nothing about virology come 2020. I just learnt it from talking to people and getting into many debates on Twitter. I started with no much more than a Twitter account and a will to find out the truth for myself. It started off a brief introduction. I was into statistics, a little bit. I knew how to analyze and data analyze statistics from university. I did a science degree, a degree in geology. And that enabled me to just, process data and process information to a certain degree. And so when 2020 came out, I started looking at some of the death statistics that they were giving us, telling us that there was this deadly pathogen that was passing between people and people, between person to person. And they just didn't make sense to me. My wife is in part Indian heritage, and we both have connections to friends and some family in India. And a friend of mine was in New Delhi, very densely packed city in India, doing charitable work through 2020. So he was dealing with people on the streets there, and the news was saying that this deadly pathogen had reached New Delhi and parts of India, but in 2020, by the end of it, had only killed something like 100 people. So they were claiming that there was this deadly pathogen in, you know, amongst the populace in India, but people living on the streets, and obviously India is very well known for having, quote unquote unsanitary conditions. He was dealing with people that were living tooth to jowl in squalor essentially, unfortunately, and he would give me almost daily updates on how people were, and they were fine. There was no symptomology of anything being passed from person to person, and disease any more than he usually saw. And I watched that and was completely befuddled by the fact that there was a complete disconnect between what they were telling us on the news and what was going on in reality. And so I started to research what was going on, to try and find the kind of causal connection between, what they were saying was happening, pathogens being transmitted from person to person. They must have done this, in science to come to that conclusion. There must be something. And this is really two parts. There's two parts to this project, or there's two parts to what I'm doing, really. If you want to learn about, germ theory and how it is built on fraud, and it does not work in the way that they do, this is the path that I would suggest that you take to start off with. If you're new to this, if you don't understand that biological that the contagion of biological pathogens is a myth, just follow as I have done, or try to investigate these areas, which is, I went on to Google Scholar and I just started typing in words, and I didn't really know what I was looking for. I didn't really know how to describe it, but it was just putting virus up people's noses on purpose. Enter, what'd you get? And it took me a little bit of time to kind of refine what I was looking for, but eventually I did find it, which was a controlled human infection model, which is where over the course of 130 years, they've been experimenting on over 100,000 people, where they have, and in the olden times, taken diseased material from people exhibiting the symptoms of, diseases that they blame on viral infection. And they have taken those and directly given them to healthy people, over 100,000 people, and taking all symptomologies, smallpox, polio, influenza, Spanish flu, all the way up to HIV and everything. Even as far back, if you really search through some of the contagion studies, they do go back as far as the plague. There are some, rough notes about people in the plague. And the common thread of this is that when you actually start to look and find these peer-reviewed, published papers that are publicly available, they all fail. Every single one. 100,000 people, they don't have a single death. They don't have bronchitis. They don't have pneumonia. They don't get sepsis. Not a single person is ever hospitalized. And my jaw hit the floor when I found this out, because that was conclusive proof. There are no contagion studies, and when you look at contagion studies, they're actually fantastic because they also rule out other quote unquote pathogens, which is bacteria, fungi, parasites. Because when you take, and the better ones are the slightly older ones, because as John Enders developed the cell culture technique, which we'll go into a little bit further on, the cell culture technique claimed to have, purified virus in a vial. And so from 1954 on, the human controlled infection models start to use the actual, virus they claim as a virus and putting that up their nose, rather than taking the infected material of people. So they slightly subverted what was going on, but in both cases, they've never managed to induce disease, even in the modern ones where they're giving people these quote unquote viruses, which do knowingly contain antibiotics. They knowingly contain, serum from the fluid around the heart of a cow, all this type of crap that they're putting up people's noses, but they still don't get sick. And this is where the kind of junction comes, is that if you really want to know and really want to understand and really want to lose the fear, that's where to start, is to start understanding truly for yourself that contagion of biological pathogens is a myth. You can see it in the peer-reviewed paper. It's unanimous. It's unequivocal. And if you then want to go and test for yourself, and that's again, something that I would encourage. Just, when just start to open your eyes and look around when people are exhibiting symptoms, when family members are exhibiting symptoms. Note just exactly how many people get sick roughly in the same area, roughly at the same time, and conduct your own studies. And that's a really great one, right? So this is where the two parts of this diverge, is because what we saw in 2020 was a scamdemic, a fake pandemic caused by fake testing, the PCR tests. Now, this kind of segues with the contagion studies, because in the more recent contagion studies, they managed to get away, despite people not exhibiting any symptoms of the disease that they're giving them. They claim to have positive studies because they come away with PCR positives. Those positives cannot mean that they're displaying symptoms, because they don't. Everybody agrees on that notion. But still, this is how they've managed to get away with fabricating a pandemic. And so this is really where I kind of ended up in 2022, was going, I was lost, because I was saying, well, all of the peer review papers, according to all halls of science, there is no contagious pathogen. But we have managed to, see some of the most totalitarian and strict political decisions being attempted to be enforced on the large majority, the large percentage of the world, and it was based on the PCR test. And so this was kind of my next endeavor, to actually go and check, whether these things were working too. Just like I checked with the contagion studies, I tried to find out about, the core principles behind how they claimed that they isolated this virus, how they claimed to have a purified form of it, and how they claimed to test its genetics, its antibodies, and whole genome sequences. And I stumbled across the work of Stefan Lanka, who had done a control studies experiment in 2022. In fact, he had released his initial kind of plan in 2016 when he stood in court. And it's quite a long story, his court case, but it over the course of two different courts, the lower court, the regional court of Ravensburg, and then up to the higher court of Stuttgart, it ended with, and I have to get this completely right, the wording: there is no evidence of the measles virus in a single scientific publication. That was the unanimous result, the unanimous decision of all three judges at the higher court of Stuttgart. And you can go, I'll put up a link to it. It's on my Twitter, feed, a thread about that. But and it's quite interesting. Essentially, the expert witness that they called up, the expert scientist, Dr. Podbielski, admitted that there was no scientific publication that fulfilled Koch's postulates. So they had to rule in the fact that there was no scientific evidence in a single scientific publication, because that's what they admitted. And as part of that, Stefan Lanka did his first control experiments, which is where he took the cell culture isolation method. I don't know, how you want to, what level you want me to start at, to go straight into explaining it, or would you like me to explain, it from the very start in terms of what cell culture is, etc.?
Maybe a brief overview wouldn't hurt.
Cool. The cell culture isolation technique is an in vitro, i.e. in the Petri dish, representation, supposedly, of what's meant to go on in your body. So they take a cell line from all sorts of different animals, monkeys, dogs, hamsters, people, fetuses, and take the cell lines, grow them out in fetal bovine serum, which is the fluid from around the heart of a baby cow, which gives it sustenance. It's a nutrient medium, so it allows this cell line to grow out because they take very small amounts of it and they grow it out. When it is healthy and when it is what's known as confluence. So confluence is just the percentage within the dish or within the plate that you're looking at when it's got to a decent confluence, covering 80 to 90% of the plate. Then they reduce the serum from 10% growth medium down to 2%, which is known as a maintaining medium, but actually, as you'll see, is not quite so clear. And then they introduce what they consider to be infectious materials. So they take snot, bronchoalveolar lavage from the chest, or mucus or blood, and they put it into these cultures. And when the cell line breaks down, when it dies, they point at the culture and say, that's because of a virus. This is the technique that was developed by John Enders in 1954 and is used as the gold standard, the only way to quote unquote isolate a virus. And so what Stefan Lanka did is he took the exact ingredients that we used. They use, in the case of SARS-CoV-2, they use the monkey kidney cell, the Vero E6 cell line. And he did not put a sample in, so that it contained no possibility of a virus. And he put the same antibiotics. He used the protocol that was used by the ATCC, which is it's essentially like a bank where you can buy all of the cell lines and ingredients for cell culture. They informally run protocols for cell culture isolation. He followed all of those and lo and behold exhibited cytopathic effect, which is the supposed death caused by a virus he saw in his cultures that could not possibly contain a virus. And so I was really interested in his work, because to me, again, this was falsifying what they were doing. And really, when you look, if you can say that you falsified that initial step, you can't claim that you have a purified virus. Therefore, you cannot claim further down the stream that you have a genetic sequence, because you don't know. There is no benchmark for what you're meant to be looking for in the first place when you're, for instance, de novo sequencing. You can claim that you can filter things out, but you can never know for certain. So in theory, if you have falsified that initial step, you have falsified it further down the line. Now, Stefan Lanka did supposedly do a whole genome sequencing. He never released that paper, unfortunately. And so that kind of led me to going around and trying to find information about this paper that didn't exist, as far as I could tell. And I found out that it just wasn't published. It wasn't published, and this is I have it from very good authority that it wasn't published because when the geneticist that carried out the work on the control cultures found out the results and the fact that they managed to assemble the genomes of numerous viruses from his cultures, that she was so scared for her job and scared for her career that she said that she didn't want it published. And that is what I've heard has happened. But nevertheless, it matters not, because I'm going to do it. I've set up this control experiments project. It's been going on for about a year now. I was running it in private to start off with, but we've decided to release the preliminary results of the experiment that we've done. And I wanted to build on Stefan Lanka's work. I wanted to take his work and to expand upon it and to really steel man what he was doing, because a few people, I saw there's a chap called Frank Visser. There's a couple more that made kind of, semi-official on blogs rebuttals to his work. And some of the claims that they made were the fact that he, quote unquote, overgrew a few of the cell lines. So we talked about, growing out the cell lines in nutrient medium, FBS, the fetal bovine serum, to start off with. If you grow it too much and it gets to the end of the plate, the cells starve each other out. So you can get kind of cell death if you overgrow them. Now, I don't think that that's what Stefan Lanka did, but we took all of those criticisms and we tried to replicate the, incorporate those into our scientific experiment design, and go further with a lot of the processes and things that we were doing to really steel man the results that we were getting. So this is the first slide. We've done this experiment 12 times. We've repeated it 12 times over 90 cultures, where Stefan Lanka did it just the once. We wanted to make sure that we had repeatable results. So if you can see at the top here, can you see my mouse pointer? I'll get this out. So here we have 10% fetal bovine serum. This is 1 times penicillin streptomycin. So we used antibiotics, and these are the lowest grade antibiotics you can get, pen strep. And we just did one flush with them with the exact volumes that the ATCC recommend to use for best practice for cell culture isolation. So we followed all of the protocols that are laid out by the ATCC to make sure that this is a standard, virological used in a virological lab, a standard practice for carrying out this procedure, so that we couldn't be accused of doing anything funky, or and we chose to really strip it back to a very basic experiment. That there's quite a big Overton window of how you the methodology that you can use to isolate. You can take it into 10%, you can passage them, you can do all sorts of things to them, but we chose to keep it as simple as possible so that you can see the results. We could have used the more toxic amphotericin and gentamicin, which are antibiotics that are known nephrotoxins, so they kill kidneys. Most of the cell lines that are used are kidney cell lines. So it's kind of strange, the disconnect, that they use a known toxin to kidneys, yet they're using and studying the breakdown and death of kidney cells, which is contradictory. So we chose to use the lowest grade antibiotics, and we also chose to use the HEK293T cell line, which is a human embryonic kidney cell. It's known as a very robust, very hard to break down, very easy to maintain cell line. It is the most widely used cell line in clinical research for those reasons. Now, we used it for 2 reasons: because it's harder to break down. So we wanted to make sure that when and if it did break down in our experiments, that would show weight to the findings that we were getting. And also, it's known as being more robust than the Viro E6, which is used for SARS-CoV-2. These are used in the HEK cell lines, are used in the quote unquote isolation of adenoviruses, lentiviruses, SV40. So they are used, just not for quote unquote SARS-CoV-2. But the Viro E6 cell line is known to be a little bit more fragile. It does break down a little bit easier. It's the one that Stefan Lanka has also already got results in. So we wanted to try something more robust to really test what we were looking at, to really, give ourselves the highest hurdle to jump over. And the second reason we used HEK cell lines is because they're human. This is again another kind of contradictory part to the virological methodology, is that they want to show their in vitro studies that are meant to be a replica of what's happening in real life, but in the petri dish. And with SARS-CoV-2, they really struggled to get any sort of cytopathic effect with a human cell line. They could only do it in the more fragile Viro E6 cell lines, in the monkey cell lines, which, if you think about it, is not very indicative of a pathogen that's meant to be deadly to humans. So we wanted to choose a human cell line to actually make their experiment better. I mean, their experiment is crap, but as get as close to it as we could with something that may give us good results, which is using a human cell line to show potential human pathogenesis. So if we look at the top here, this is essentially our negative control in this experiment, because the independent variable is not there. It's never been there. There is no independent variable in the initial isolation method, because it's a whole load of gunk, sputum, or BALF, or mucus, and so they've never kind of purified it to start off with. The independent variable in this is the fetal bovine serum and or the antibiotics. So what we're doing is actually nothing to do with viruses. We are just looking at the cell culture technique, and we are falsifying this technique. So this is the negative control, a 10% fetal bovine serum culture. It's happy, healthy. It's grown to a confluence of 95% in this. So it's right there. You can see here cell viability, 95% dead, 5%. So we have sought to get objective verification with this. Now, a lot of the times in cell culture isolation, they just use the opinions of the virologist that's conducting the experiments, whereas we wanted to make sure that we weren't being accused of just subjective opinions and take that away. So we used what's called COUNT-S. It's laser spectrometry. It's a cell viability counter, essentially. It just shines laser, bounces back and shows the transparency, the opacity of the cells that are in the dish, and can give you a readout and an objective opinion of how many cells are viable within the dish. And so here we can show adequately that there is no overgrowing. So it hasn't reached 100% confluence. It's a very good, happy, healthy cell line with a little bit of room to move, but a very great place to start so that we can show that the CP that occurs in the cultures is starting from a very high benchmark. So here we have the first parts to our control, where we've reduced the serum. We've reduced our independent variable down to 2%, which is supposed to be a maintaining medium. But as you can see, lo and behold, we have these huge gaping holes, which are cell death. They're CPE, cytopathic effect, apoptosis, cell death, or cincture, which is where the cells clump together. And you don't need to be a virologist to see that these cell lines are dying. They're dead. They are indeed, up to 40% dead. Yeah. In objective verification from COUNT-S. Here we also see that you can put 1, 2, and 3 times the antibiotics as you go along, again, according to ATCC. But we wanted to alter these to see how much cytopathic effect was caused by this, whether it was just the reducing of the serum, or whether it was in combination with the antibiotics, or whether it was just one more than the other. And as you can see, it slightly raises as you put a little bit more antibiotics in. So starting off with 32%, it kind of comes down a little bit, but these readings vary slightly across the whole plate. What you're seeing is a snapshot that's a very, very tiny percentage of the whole plate. So you read and it gives you a rough estimate, and so you can kind of estimate across. But if you get to the last one of 40%, you can see that there's a slight increase across the cultures. But the main difference, obviously, is the removing of that serum. It is starving the cells. It is known to starve the cells because you're removing the nutrients that you got to grow it in the first place. And that's what's occurred. That's what's occurred in every single cell culture that we did. Over 12 experiments in 90 cultures. We're slightly different. We vary slightly some of the materials that we use. We use a few different. We use amphotericin in some parts. We try to, and here we're looking at reducing it down to 1% fetal bovine serum. So we go slightly off protocol here for HEK cell lines. They don't really go down to 1% fetal bovine serum in a lot of the protocols and a lot of the methodology, but we wanted to just have a look and see if it made that much difference. On the top here, you see, and sorry, I didn't say this is at day 4. So all of the all of the plates that we took out from incubation at day 4 to observe and put them under 20 times magnification. And day 4 is as you will see, a very standard time to allow to observe these cell cultures in. So it can be up to a few weeks. In some of these protocols, they're leaving these cell cultures for. I've seen one that's been left for nearly a month. Okay, a month before these things have broken down. So 4 days is pretty rapid. So we compared 2% to 1% here, and as you can tell, 30 to 36, it's actually a little bit more, 32 to 35. We're in pretty much the same ballpark here. You're not seeing a hell of a lot of difference. So it suggests that the fetal bovine serum, the initial starvation coming from 10% down to 2% is what does the damage. Once the nutrient medium is depleted, then it matters not that it's gone. The cells are already dead and dying. So we just showed that within protocol, from that 10% to the 2% when they supposedly infect the cell culture, is when the damage is done. Now, I mentioned that we had the negative control of the 10% fetal bovine serum to show that to show that we are controlling in our own experiment. We don't want to be guilty of not having the controls. And I'll just go into this because obviously there's a small claim that virologists do negative controls. They do a thing called mock controls. Well, on the surface of it, you see that people, virologists, when you read some of them put the pictures up of the mock controls they do. Not hardly any of them. It's a handful that you actually see, the cell lines. You see the results of the mock controls. I've interviewed hundreds of virologists on this, and I have got quite a few of them to admit by proxy that they are not doing negative controls. They are doing weird and wonderful things like PCR testing and throwing away, or keeping them in different environments, i.e. they're remaining in 10% fetal bovine serum and not reducing the serum to give the exact same conditions that they're doing the infection in, which is not a control. They will claim that they're doing mock controls, but you have to really pick a way to try and find the protocol for how they're handling these controls, and inevitably they handle them differently to the infection things 100% of the time. I have never seen a full methodology written for a control in any single virology paper. They never have put out exactly what they are doing and stipulating that they're doing the same. It's all just based on wink, wink, nudge, nudge, they're definitely doing it. Yeah. And when you actually get to the nitty gritty, and I have worked, with this project with people who have described how in industry these scientific experiments are conducted. And it was eye-opening to me to actually hear it from the horse's mouth that what they describe in terms of doing an experiment is thusly: that they know what they're looking for before the experiment starts. They know what they expect to see before they even start doing the experiment. And so if the experiment doesn't do that, then they change the methodology, they throw it out and they start again. And they tweak it and they move within this giant Overton window that they have to get the results that they've imagined in the first place. And on the surface of that, when you actually view it like that, what they are carrying out is scientific fraud. Now, I wouldn't accuse them of scientific fraud because I just think that the entire thing is kind of built up around people not questioning what they're doing. They think that what they're doing is just best practice. Oh, I must have fudged up somewhere. Not that the results that they're getting show that the process that they're doing is bullshit, but the fact that they think that they've mucked it up somehow and they need to adjust to get the results that they need. So I don't think that these people have bad intentions. I just think that they've been taught, they've been quote unquote indoctrinated to carry out these processes blindly, and the results that they're getting are actually not indicative. And as you can see, they're quite clearly not carrying out proper negative controls because we have done it in 90 cultures and exhibited not a small amount of cytopathic effect. These cells are half dead. They're gone. Yeah, just with the most basic, the lowest grade antibiotics. So, as you can see, the call or the claim that mock controls are adequately done is quite clearly false. So here we're talking about the negative control. If you look at the bottom here, there is a problem with using a positive control. Now, quite a few people have suggested that you could use a positive control to compare the cytopathic effect of, the viral titers that they have. That you can go back to the ATCC and you can buy, what's quoted as virus, right? Why don't you put virus into this and see? Well, there's a very easy reason why we don't, is because the ingredients in those virus titers contain falsified ingredients. Okay, so they only get those quote unquote purified virus titers from cell culture. So that cell culture knowingly contains antibiotics, it knowingly contains reduced medium, and both of those things both cause cytopathic effect. So if you're going to test it out, you have to knowingly admit that you are adding things that cause cytopathic effect. So even if you added it and it had way more, I mean, personally, I don't think that it would have way more. You would have to have the cell line disappear in front of you, to have more than 40, nearly 50% cytopathic effect in 4 days. I don't think I've seen a virology paper with more destruction than that, really. You can get towards like 50 and 60%, but really, you're taking it out of incubation before you get there because you're almost having no cell line left to do anything with. And so, sorry, just knocked my microphone over. And so I don't think that even if you put these viral titers in, I mean, I am going to do this as part of the control studies project because actually we want to take those viral titers and we want to put them under electron microscopy to have a look at them to see if they contain all the same particles, and we know damn well that they won't. But I think that that'll be an interesting, look to something to investigate. But that's the reason why we don't put them in as part of the positive control, because they knowingly contain substances that cause cytopathic effect that are not a virus. They contain the reduced medium and the antibiotics. So we have developed what is the closest to an adequate positive control, and I'll say this kind of tongue in cheek. We used asymptomatic sputum, so sputum from an individual that was exhibiting no symptoms. They were asymptomatic. We diagnosed them being asymptomatic because when we put their sputum in the cell culture, lo and behold, it exhibited cytopathic effect to 35%. So yes, that may be a logical fallacy and circular reasoning, but that's virology, unfortunately. I don't write the rules. Don't shoot the messenger. So we used sputum. We put that in there. there to essentially we are, contaminating, quote unquote, contaminating our own cultures to look to see, if it induced a lot more cytopathic effect, because it would be interesting if all of a sudden, again, it just went off the charts and we saw something completely different. But as you can see here, looking at the top slides, we have 34% dead, so 34% CPE. And at the bottom, with the addition of the sputum, we have just 35%. So it's almost identical. We're seeing absolutely no change from intentionally putting asymptomatic sputum in. Now, that sputum, if you read virology textbooks, should contain millions of little viruses in them. We are bathed in a sea of viruses, according to the priests of virology. But yet it's not causing any cytopathic effect any more than with no possibility of there being in there. So I just want to clear this part up as well, is that I can say that there is no possibility of a virus being in our cultures, because when you buy the cell lines, you buy them from the ATCC, they come with a guarantee of non-contamination. So this is how we start. This is how we can prove and we can falsify this as an experiment, because when you buy them, they come negatively tested, they come sterilized. And so what you have is a blank canvas to start with. If these things were contaminated with viruses already, they would be useless for the entirety of clinical research. You would have cell lines everywhere that would have virus in, and so you wouldn't be able to research on them, and the entire world of clinical research would dissolve and become moot. So the claim that they could be contaminated is moot, because by their own confirmation methods, they are free from contamination. The same with the fetal bovine serum is inactivated, and antibiotics is there to supposedly take away or it does take away the bacterial, fungi, and parasite, bacterial and fungi contamination. So here is from the American Society of Microbiology. This is in the cell culture handbook of best practice and what to look for. And I'll just read this out to you. The rate of CPE, cytopathic effect appearance, is also a characteristic that can be used to help identify viruses. In general, the rule of thumb is that a virus is considered slow if cytopathic effect appears after 4 to 5 days in cultures inoculated at low MOI. MOI is just how much virus you put in there, but we had 0 MOI. And rapid if CPE appears after 1 to 2 days in cultures inoculated at low MOI. So in our cultures, we had cytopathic effect appear at 48 hours in 2 days. And by day 4, when we actually observed and we took the images, it was nearly half dead, 44% cytopathic effect. So according to the American Society for Microbiology, we have a virus in a dish that cannot possibly contain a virus. So we have falsified the cell culture isolation process, not once, but 12 times in over 90 cell cultures. We have shown that the indicative thing, the cytopathic effect, the observable effect, is made by the materials alone and nothing to do with the possibility of a virus or any pathogen within a sample. So here, just the bottom left, just to back up what I was saying, the HEK 293 cells are generally regarded as a robust and low maintenance cell line. And another thing to look at, just, of interest, when you start to scratch away at the protocols within these things, it's kind of funny, the cell lines that they use. So here's another one that's pretty standard, used in isolation of quote unquote viruses, is the HUH7 cell line. It's a permanent cell line established from a male hepatoma tissue, which was surgically removed from a 57-year-old Japanese male in 1982. And that's kind of the same with nearly all of these cell lines, is that they're not taking cell lines from healthy, strong tissue, from the bicep of Arnold Schwarzenegger and trying to, break it down. They're taking it from this is a 57-year-old Japanese male who was dying of a kidney tumor, a cancerous tumor, on his body that was killing him, that they've taken and frozen since 1982. And they've kept it frozen, and then they unfreeze them and grow them out, and then say when they die, these already diseased tissues, that this is indicative of something killing them. You see them from, obviously we're using them for, unfortunately, from embryos, from fetuses as well, tiny fragile little beings. They have hamster kidneys, they have dog kidneys, monkey kidneys, and quite a lot of lung cancer tumors as well. So it's not really a fair fight that they're setting up in these supposed in vitro studies. They are rigging them from the start by having diseased and dying tissue and dying cells to start off with. So that's kind of where we deviated from Lanka's work. In essence, we knew essentially what we were going to get before we started with that, because it's very obvious that tissues break down if you starve them. Yeah. So we were confident before going into that we would receive cytopathic effect. We didn't know that it would be that bad, but we decided to go that one step further, and we wanted to take a look at these cultures. So we sent these cultures off, we boxed them up, we sent them to another CRO. We sent them from one CRO to another via recorded scientific posts, so on dry ice, all to make sure that all of the processes that we were doing, it was all done under flow hood when we did all the experiments. So we've documented everything in terms of contamination and chain of custody for taking samples and moving them around and sending them to make sure that we're not contaminating them in any part. And we sent them off for transmission electron microscopy. Now, we didn't at the time, because it was privately funded, we didn't have a lot of money to put towards the experiment with the transmission electron microscopy. We were kind of getting a bit thin, and so unfortunately we had to choose a slightly lower package, because if you get them to look at a sample that could contain a virus, then they kind of double the cost, because they need to do it in the hazmat suits and all that bollocks. So we just we got them to look for extracellular vesicles, because we knew that they would be somewhere within the breakdown of the cell line. And also we didn't have a chance to actually direct them where to look, where to take photographs. So bear in mind that these are photographs that they have selected. They only selected 6 photographs, okay? 6 photographs. This is the first time that we've done it. It's actually a world first of looking and doing a control study experiment of an uninfected culture to compare and contrast. So it's a world first that we did here, did in this experiment. And we got the CRO to look for and identify extracellular vesicles. And this is what you see in front of you. So you can't see the bar graph off to the side, but I can tell you that it's about 2,000 nanometers, which is way much bigger than a virus. You can see that it's empty. You can see that it's kind of misshapen. It's quite clearly not anything that could be described as a virus, which will come in handy a little bit later, because we can all categorically say that's not a virus. They've identified an extracellular vesicle. Here on the top screen in purple, and a few of the other, I'll just get the pointer up again. In the purple square is what the CDC consider is an Omicron subvariant BA2 SARS-CoV-2 particle. These are some of the particles around it that are also SARS-CoV-2, according to the CDC. So if we describe it, and this is what we're going to do with the cell culture, we're just talking about the cell culture. There's no other indicative thing. We are moving through each of the protocols, each of the methodologies that are claimed to show evidence of existence of a virus, and we are going through and falsifying them one by one. So what we will do is just talk about the physical look, the physical appearance of these particles that we're seeing. The CDC says that they're round, that they have some kind of slight coating around the exterior. They are contain these inclusions, which are dotted in a circular fashion, and between them, here we have maybe 4 or 5 of these inclusions in that crescent format here, whereas here we have maybe about 12 to 15 in the circular format. So a slight variation, even in the particles that they're analyzing. You can see this one's slightly bigger, this one's slightly smaller, but roughly they were all round. They all have a coating around the outside. They all have these protein inclusions, and they're all in a spherical pattern. Here is our culture. From a culture that cannot possibly contain a virus, we have a round particle. You'll see that it has a coating around the outside. That coating is slightly darker, but that's just to do with the brightness, the contrast. We got about 10 different contrasts to look, and I just picked out the best, the sharpest image for the contrast. So the actual grayscale is slightly darker in most of these, but they contain this protein inclusion, and those protein inclusions are in a spherical format. I can also tell you that this is the same image from previous, is that they are exactly or very, very close to 100 nanometers in size. I've taken, this is the whole image that we received from the laboratory. You can see the bar scale down here says 500 nanometers. So what I've done is I've cropped it out and I've kept it the same ratio of scale up here, and I've put them next to each other. And if you could see that goes in roughly about 4 to 5 times. So you're looking at a particle which is 100 nanometers in size. And here from the National Institutes of Health, it states diameter of a SARS-CoV-2 particle varies from about 60 to 140 nanometers. So in our control cultures, we have a particle that looks exactly the same shape, exactly the same size, contains roughly the same amount of protein inclusions in the same spherical pattern with a coat around the outside. So here we have on the top, what the CDC claim is a picture of a measles virus. It's oval. Again, we'll just describe it, purely on its physical attributes. It's oval. It contains what they label as a matrix. Particulate interior. It contains dotted proteins. They label these as phosphoproteins or large proteins, but they are sporadically dotted around the particle. It contains a viral membrane, and that's it. It's 250 nanometers in size. So on the bottom here is again from our culture. And if we can just describe what we're looking at, again, it's oval in shape. It contains this matrix, some kind of fluid that's inside the oval particle. You see the proteins, or what could be labeled as proteins. They are sporadically dotted around, and you see it's encapsulated by a membrane. And again, if we go and we look at the original image, so the original image is here, the bar graph, and I've moved because in terms of identifying, these particles, the main thing is size, because as you can tell from the bottom image, these things are much, much larger than what, is this, this thing here is much, much larger than what could be considered a virus. You're talking like five times the size. Yeah. There's lots of things in there, but there's also particles that are oval in shape. This is exactly half of that bar chart. So you're talking 250 nanometers. And if we read off what the CDC claim is the size of a measles virus, it is 120 to 200 nanometers in diameter. So what you're talking about is a particle exactly the size of a large measles virus. And there's more. In the top image here, we see what the CDC considered to be HIV virus. So again, we'll just go and we'll describe what we see in front of us. We see, and all of these are roughly rounded. Some of them are slightly misshapen, but roughly rounded particles. They all contain this outer coating. They contain a nucleated protein mass in the middle, or a fried egg type thing, and then a small amount of matrix in it. You see here, the bottom is a picture from our cell culture. And again, let's just describe what we can see. It's a roughly rounded particle. It has this kind of matrix in it. It has a membrane around the outside. And most importantly, it has the nucleated mass in the middle, of fried egg, type shape. There's also a couple more in the image that you can see. And so again, we'll go back to the original image where we pull the bar graph up and we pull it up to the size of the image, and we can see that fits in 5 times. And so we'll read again from the National Institutes of Health. The mature HIV particle is round, measures approximately 100 nanometers in diameter. So again, we have a particle that is exactly the same size, exactly the same shape, exactly the same nucleated protein mass inside, and the matrix. So with this transmission electron microscopy, this is from 6 images. All of those images that you saw were from the first 6 images we ever had done. And we had 0 control over which images to look at. And we found and identified and cross-referenced by all of those metrics, HIV, SARS-CoV-2, and the measles virus, supposedly particles that could be identified as those within our control culture. And we could get a package that enables us to look and choose. We get an open microscope session where, when they mount the slide, they give us a Skype call, and we can go through and we can direct them as to where to look. And we have absolutely no we have no doubt that we will be able to find every single virus known to man, in a culture. If we had the chance, in impeccable detail, we could find clusters of them. We could find anything that we wanted if we were to be able to direct them around and have a look and do even more, because in just the first 6 images that we've ever got, we managed to cross-reference those. So we've released these results, and the part of the project is that we take firm rebuttal. I take firm rebuttal. I want to hear people's criticisms of it so that we can adjust and make our scientific findings that stronger, and for people to engage in what we're doing. Unlike what virologists do, which is all behind closed doors, it's all unquestioned, it all goes unchallenged. They release it, they peer review it, and it's done. There is no questioning it. We want to be different, and we want to open it up to the floor. And so I've been engaging over the last few years with people on Twitter that are they're claimed professional working microbiologists and virologists, and I've interviewed a lot of them about Lanka's control studies. That's how we came up with a lot of the methodology that we use and the protocols, and some of the things that we've carried out. And we wanted to find out about what their thoughts were. So here we have K-State Turk, who's a microbiologist in the US. He says TEM is not an identity test unless it's coupled with immunostaining. Spend your money on mass spectrometry and genomic sequencing instead. Virology Truth writes, the lab states themselves that those particles could not possibly be extracellular vesicles. If they weren't, what were they? He says, I need to see the protein composition of these particles. He's not showing the data because he knows it'll make him look foolish and make him look farcical. That's not true. We haven't done the proteomics. When we do the proteomics, which we are going to do, the control studies that are coming up, we certainly will show them. Val says, and this is the crucial part, what else could those particles be? And K-State Turk says, I don't know. That's why you need an identity test. So here's the crux of it is that there is no explanation for what these particles are. Yeah, the fact that we can cross-reference them so accurately with what the CDC claim they are, there is no explanation. Oh, it's not a meso-viso exosomic thing. I have no idea how they got there or what they're doing. Yeah, the only way that they can clarify is by taking it away and doing PCR testing or antibody testing. He suggests immunostaining. So just when we're just looking at the transmission electron microscopy, we have adequately falsified the fact that they cannot tell. They need other methods to claim that that a virus exists, essentially. So here we have Dr. Thomas Moore, again, another microbiologist based in Switzerland, I think he is. He says, it's absolutely logical. They observed cytopathic effect. So what? In virus-induced cytopathic effect, one can detect the proteins in the genome of viruses. They didn't detect that. That means the experiment was a huge waste of money. Well, again, we haven't done any of the genome sequencing. We're currently doing that at the moment. Brendan Tech writes, what virus caused their CPE then? Because obviously we have no virus in the culture. And Thomas Moore says, CPE is caused by many things, starvation, for example. Now, it's very well known within the industry that when you starve the cell line, there are plenty of papers, all published, all peer-reviewed, all put out, that logically, when you starve a cell line, when you reduce its nutrient mediums, the cell dies. So when people are being intellectually honest, they will say that there is no proof of a virus after a cell culture isolation. You need everything into the PCR. The PCR is the Custer's last stand of virology. It is where all of their eggs are within the basket. And that's why we have gone public with these preliminary results to show everybody that this is where the game is. And Strand here, Thomas Baldwin, who's a plant virologist, says, do you take that bet? If Jamie's mistreated cells, they always have to get a dig in. He believes erroneously that they're mistreated. Produce a genome that resembles a novel virus, virology is falsified. So this is where we're at. Everybody admits that we're standing at the precipice, and the last thing to falsify is genomics and PCR. So here's where we are. Here's where the project has been brought up to. So have further studies, the PCR, the full transcriptomics, the antibodies and proteomics. The next steps, we're planning to put together a lab with a thermocycler and core lab equipment in order to test PCR samples and software, as we're unable to send off the exact controls we want. Within the PCR, it's quite difficult because we want to control by sending in some pretty funky samples and doing some things to try and there's so many moving parts within PCR. We want to send off like just trying to put the positive control in, just trying to put the primers in and nothing else, just the sample and no primers. And you can't really send those into a CRO because they won't, they will start to ask questions, and we have to do everything blinded. We have to do everything blinded because we don't want them fudging the experiments, if they know what we're trying to achieve. And so really to falsify the PCR part exactly specifically, we have to kind of put a lab together to be able to do that in our own remit. We can go, and in the short term, we're doing full transcriptomics, because if we falsify full transcriptomics, essentially, even a PCR positive, they cannot definitively tell you that the virus is in there because it's only a very short sequence. They can only say, we think that it's in there, but they have to clarify with full genomic sequencing. So if we falsify the genomic sequencing further down the line, the PCR becomes moot. I would like to do the PCR just on its own, but we're kind of jumping ahead to go back, if that makes sense. We would like to do further the first two observations experiments by buying more cell lines. We want to try in every cell line, as we said, the HUH, just to have a look, the Vero cell line, just to show that all of these cell lines break down into starvation and to see how they react. We would like to offer the chance for people donating to propose more control experiments to perform. This is an open and live experiment. We want people to be able to engage with what we're doing, to get on board, to understand the processes as well. This is a decentralized, totally crowd-funded and totally crowd-sourced experiment that we want people to participate in. We're not, doing it behind closed doors like virology experiments are done. All of science is done, behind closed doors like this. We want to open this up to the floor, and we are doing things like, the new part of this is to show an adequate methodology for people, for the layman, for the public, for anybody to see the merit in what we are doing, understand and come to the conclusion in their own mind that what we are doing represents good science. And so what we're doing is we are going further in our methodology, which is to take video of the methodology. Okay, we're going into the labs and we have videoed the cell culture taking place, for instance. We have videoed, them taking the pipette and moving around, all done within a flow hood, all done. So all of the questions of contamination can be satisfied, all logical questions that may arise from just the bystander of saying, is this adequate science? Does this make sense in my mind? That's what we want to satisfy, not peer review, because the peer review process is corrupt. We're not looking, I don't want these results peer reviewed. In fact, if a publication asked to peer review them, I would say, no, thank you. I don't want anything to do with that system. What we're doing is much, much greater. We are satisfying the logical mind of the greatest and most critical peer review, the people. We have done full video. We put up every single minutiae of detail, the flow hood used, the HEPA filters within these things, the amount of volume of wind that they're moving to satisfy every single part. These aren't done in peer-reviewed papers. They just expect you to believe them. And so we're going further. We want to make sure that this new way of conducting science, a decentralized science, is actually beneficial for people's education and actually finding the truth of what is an agreed upon truth. So what we're sharing with this project, all the methodology and the materials, the chain of custody, the sourcing of materials, all of the codes, the barcodes, where we got them from, will be written up in a scientific document that we are making open source. So we are making all of the results public for free. We're giving everything away. We're doing video explanations on how to use these documents, so what they mean and how to represent them and keep them as yourself. It can be used, for example, as the likes of Lanka and Marvin Haviland, and all the others winning based on the no virus claim in court. As I explained before, Lanka won, getting them to admit that there was no scientific evidence of a measles virus within a scientific publication. Marvin Haviland won his court case. He got over 30,000 euros worth of fines for not wearing masks. He was quite comically going up and in the streets of Germany and kind of tapping policemen on the back of the shoulder and saying, "Look, I've got no mask on. Can you give me a ticket?" And he was ratcheting up all of these fines, and eventually got into court and stood on a no virus claim, and they dropped his case when they realized that actually, he knew what he was talking about. And so you don't hear about these wins in court based on no virus too much. Unfortunately, there are a lot of well-intentioned people during 2020 that tried to challenge the government on things like lockdown, mask mandates, vaccine mandates. And unfortunately, a lot of them, I mean, notably, there was a guy called Simon Dolan in the UK, who I follow quite closely. He runs a private jet company and had the fortitude to go and challenge the UK government on lockdown. And unfortunately, he lost based on quote unquote asymptomatic transmission being a thing, and when we kind of look back at it and go, it's a kind of eye roll thing, but these are part of the processes that we have to go through to, and really where the proper meat and veg of the the legal scientific vehicle for proof is within control studies, within the control. That is the scientific vehicle. And so we have very good standing on what we're producing. And we want to teach people how to use them and how to back themselves scientifically and legally if 2020 ever came about. Because again, I'm based in France and you're in Canada, and our, governments were extremely overzealous, extremely totalitarian, with the politics that they put out in 2020. I had the vaccine passport here for nearly a year, and my children were banned from museums and restaurants and public soft play centers. They were banned from nearly every part of society for nearly a year, based on these ridiculous and authoritarian political mandates, or whatever you want to call them. Who knows what they were? But that's what we want to stop, ultimately, is by giving people the power to and enabling them to back themselves. So if you meet if you try to travel internationally, so I'm in France and I had to go back to the UK. I wanted to go back to see, my family during 2020 to 2023. And, unfortunately I had to do the PCR shit thing where you shove the thing up your nose, because there was seemingly no other way for me to get back. And, having to do these fucking tests, to get back. And if I had had the results, if I had had these documents to be able to show, look, you have no proof, the PCR test means nothing. I can back myself legally, for getting past and getting through these problems. In school, they're trying to force, childhood vaccinations and so on and so forth. So this document can be used in numerous ways to legally and scientifically back yourselves. So this project has very kindly been hosted by Alex Zek at The Way Forward. If you do feel inclined to donate to it, that's really, really helpful and very much appreciated, because the more that is donated, the more control experiments we do. We've just sent off for the genome sequencing 4 samples. I put out the receipt to the CRO to show that we are doing it. The quote that we had from the CRO, and that's $6,000 for the 4 samples. So it's quite an expensive hobby, this. So any donations would be very much appreciated, and they just go into more and more science being done, and more and more controlled experiments being done, and for growing and hopefully towards making a lab so that we can not only falsify the PCR, but actually more importantly, we can go a little bit further. And because one of the things is that we have no doubt that once we get to the end of kind of falsifying this, they will just tweak a few of the elements within these protocols and run them again. That's what they do, with drugs. Whenever a drug gets done for, causing harm, what they do is they take it off the market. They change a couple of the things, a couple of the chemicals in it, and just re-release it. That's how they get around these things. And so really we have to be in a live situation where we have a lab, so that if they run, for instance, with the bird flu bullshit, we can just take their exact protocols, their exact methodologies, just run them through our controls and show them that they're talking shit again. But we have to be in the position to be able to react, so that it maybe puts them off trying a lockdown or trying all of these things. So we would like to be in the position where we can react quickly. So that's it.
Amazing.
There we go.
Well, thank you very much for that presentation. It's unbelievable work what you're doing. We'll definitely put those links down below so that all listeners can hopefully directly go right to that page and share what they can, because this is truly this is what's really needed in this time. So how can the listener follow what you're doing? Is Twitter the best place? You seem to be quite active on there.
Yeah, Twitter's good, at Jamie _a again, because I got banned so many times. But I'm releasing all of the results. Writing a lot more long format stuff on Substack. So that's substack.com/at control studies. That's it's a bit more highbrow. I get into all sorts of tussles on Twitter. It's just dealing with loads of people, but it's a little bit nicer over on Substack, longer format, and all of the results will be the methodology, the materials, and the further results that we get are all going up on that Substack. So
amazing. We'll put all those links down below. Honestly, I'm following along real close to this. I think this is just absolutely amazing. It's what's needed. It's just amazing. So thank you for everything
no,
that you're doing.
thank
And
you,
for
Liv, and thank you for giving me the platform and the time to go through it. And it's great to have your support, and I'm excited too. I just wanted to know the truth and be and see what it was. I didn't know if I was kind of barking mad. You kind of sit on Twitter and you go, oh, and you hear about, oh, the virus has never been isolated. You hear that's the hook word. And you don't really know until you actually see. And so that was some of the surprising things for me, is opening the transmission electron microscopy, and we had absolutely no idea. And so it was kind of fun. It's like a high school project again. It's good to get that kind of excitement about science, I guess, in a kind of geeky way. But I've been inundated with positivity from people that have had the same feeling, that they just want to see for themselves. They want to see the truth, and that's what it is. It's just the truth, and no more and no less. And I think it's important to bring to people, and it's a positive experience.
And I'm eager to have you back on, even to just discuss sort of the philosophy behind what you're doing. I think that you'd have lots of insights there, because, what we're getting back to is true science. We're not concerning ourselves with the profession of science with vested interest and things like that. Like, I started off same as you. I was just curious, and I thought, I was quite on the opposite side when I was studying this sort of material, and I woke up every day to try and disprove the claims being made by the terrain community, and in that, I just proved it more and more true, and it kept disproving the germ theory. So it was just a
Yeah.
pure curiosity in my search. And I think that's true science. And I think we're seeing a resurgence of true science. And you mentioned decentralized science. That might be a great method, science for the people. So I just
That's right.
absolutely
I'd
love what
love
you're
to come
doing.
back on and just do a show about that, because again, the Substack is kind of half about decentralized science projects. I've talked about this in private, given private presentations to people for the past year on small groups of setting up other control experiments. I've got interest from small groups in Japan, in Venezuela, in South Africa that would all like to start their own control studies. How they meet CROs, how they engage them, just as laymen, because it's totally possible. It's totally
Absolutely.
possible to conduct all of this science as a layman, as crowd-funded science, and using the agreed upon um, methodology of an accuracy that you see within contract research organizations. Essentially, Mr. Borla, Albert Borla, from the CEO of Pfizer, is just contracting these CROs. He's a businessman. Actually, his degree is in his degree is of he's a veterinarian, right? He's he's not doing the science himself, right? He's paying other people to do it, and so all we're doing is we don't need to change any huge parts of the infrastructure of what's already occurring. It's just a bottom up approach rather than a top down. It's just kind of turning it on its head and being for the people. And I would love to, explain more about, the philosophy behind decentralized science. And, maybe I could, and we were again talking a little bit off screen. I'm in France and you're in Canada. Maybe we can do small excerpts in French. Because I know that there
Definitely.
are, there is a little market out there for other languages, for getting the message out there of no virus and contagion being a myth. So
Yeah.
maybe we can expand on it.
I would love that. Awesome. Well, thanks again for today. I really appreciate your time.
Cheers, Leo.
And I want to thank you all for listening. You should all know that it's not medical advice or scientific advice of any kind, used for your informational purposes only. But also remember that we are all responsible, sovereign beings capable of thinking, criticizing, understanding absolutely anything. We, the people in the greater forest, are together self healers, self governable, self teachers, and so much more. Please reach out if you have any questions, criticisms, comments, concerns, whatever it is. I'd love to hear your thoughts on this episode. You guys know where to find me, Beyond Terrain. I'm on Twitter a little bit more now too, so if you want to holler there, and I know Mr. Andrews is eager to chat about this stuff too. So um, just reach out with anything you got, and if you took time to listen to the whole episode and you enjoyed it, give us a like, share, comment, subscribe. Sharing's the best way to help this channel get the word out. That's really what we're here for. So that would be fantastic. You guys could do that. All right, guys, thanks for listening. Remember, there are two types of people in the world: those who believe they can, those who believe they can't, and they're both correct. All right, guys, take care.